Okay, thank you very much for inviting me to this talk. The title of this talk is Protein Threading Using Context-Specific Alignment Potentials. So first, the outline of this talk will be divided in 5 parts. The first is where we are at the template-based modeling, and the second part is what's our work has already done for these problems, and the third part is what's the problems occurred in our previous work, and the fourth is our solution in this paper submitted to ISMB 2013, and finally I just introduce everyone to welcome to our server. So let's just briefly introduce the template-based modelings, or someone called the threading. So currently, template-based modeling is the mainstream approach in protein structure predictions. This is because on the observations that although we have around the 15,000, can you see my mouse? Okay, 50,000 non-redundant proteins in PDBs, but we have only about 1,200 unique structures in scope. And you see these figures from the recent years, the number of the new fold has not appeared a lot. So this results in a fundamental assumption that we could use the already known structures to predict the unknown new sequence. So more formally, the definition of template-based modeling is given a query sequence as well as a template database, and we align each template and each query to find the best match, and upon this best match, we build the 3D structures, and this is our mission for template-based modeling. So let's introduce some related works according to the appearing time to the market. First, at the beginning of the 1990s, scientists found that similar protein sequences would lead to similar structures, and this results in the template-based modeling purely based on sequence alignment. And the most famous one of them is BLAST and FASTA. I think most of you will know it. And this method works for close homologs, which the sequence identity between the template and the sequence is above the 70%. So next, in the end of the 1990s, scientists found that although some protein sequences are not so similar, but these sequences have the similar profiles, which the profiles is defined as a matrix that represents the multiple sequence alignments of homologous proteins. And these similarity profiles would also lead to the similar structures due to the evolutionary conservations. These great findings result in the profile-based methods such as SciBLAST, HMMER, and HHPRED. This greatly enhance our ability to template-based modeling, since now our sequence identity threshold could be lowered down to 40%. However, at the beginning of the 2000s, many further examples has been discovered that although the sequence and the template, they share very similar structures, but they don't share even the similar profiles. So for tackle these cases, scientists have incorporated the structure informations or the context-specific informations into the sequence profile-based methods. These methods, for example, such as the Mooster, Raptor, and the CSBLAST, can work for distant-related proteins, which the sequence identity between the sequence and template is less than 40%. But the remaining challenging thing is that how can we utilize these heterogeneous and complex features including the structures, sequence profiles, and as well as to increase the alignment accuracy. So, in order to solve these problems, we transform the protein threading problems into a machine learning problems. As we know that the machine learning problems has the three main parts. The first is, what is the data? The second is, what is the model? And the third is, how to design the features. So to provide high-quality training data, we develop our in-house tool called Deepline, which is a protein structure alignment tool that employs a versatile scoring functions that incorporate evolutionary, hydrogen bonding, local substructural similarities, as well as the spatial proximities beyond two input structures. This software can generate the structure alignment as accurate as the human expert manually created ones. Second, we design a machine learning model which is called the CNF, which is the abbreviation for the conditional neuron field, which integrates the powers of the conditional random field as well as the neural networks, which it can simultaneously model the complex and heterogeneous features using a nonlinear function instead of the linear function appeared in the CRF, and it can also exploit the interdependencies among adjacent positions, which is not appeared in the neural network. So here we move into the first part, that how to define the label for protein alignment paths. In details, we transform alignment paths into a series of continuous labels with M, I, S, and ITs. So after the definition of the labels, we could apply Deepline to generate the training data, which is from the structure alignments. And I just briefly introduce about the Deepline. The Deepline scoring function consists of the two parts. The first is the local similarity, while the second is the global similarity. The first local similarity is incorporation of the Blossom matrix as well as the Cleason matrix. The Blossom matrix will describe the evolutionary informations among the two input proteins, while the Cleason matrix will describe the local substructural similarities described in conformational letters. And after the two proteins are supervised together, we can use the VIJ, which measures the angular similarities. That the purpose for this is for the hydrogen bondings, and the DIJ measures the spatial proximity, which we can use the RMSD or the TM scores to measure the similarities. Because these components are considered by the human experts when doing the manually curated structure alignments, so our automatic structure alignment tools can generate the alignments which is similar as the human expert generates. So after we described the data part, we then move to the model part. And to describe how to use the CNF to model the protein alignments. Basically, we define the conditional probabilities for a certain alignment A, given template S, as well as the sequence S. That is like a Boltzmann machines with exponential families and normalization factors here. So the training purpose is simply to set the parameters in this function E that we to maximize the total probabilities for our training data. That is the structure alignment. The function E itself consists of 9 different neural networks. Since there are there are 3 labels, and the adjacent one of them is 3 times 3 is 9. for each neural networks, we can, for example, for this sample is with the state is M to IS. We can use a window of size 11 in each aligned positions to feed the features to the neural networks. So in summary, CFPRED is actually a context-specific alignment model which could calculate the alignment probabilities. Finally, we will introduce the features we use to our machine learning model. We can categorize the features into two parts. The first part is the sequence-related one. The second part is the structure-related one. For sequence part, we use the how similar the two residues is, and how similar the two aligned positions profile is. For structure-related ones, we are using the 3-class and 8-class predicted secondary structures. And we also use the predicted solvent accessibilities. Finally, one thing need to be pointed out is, because we know there are many disorder regions in the proteins, and also these regions can be predicted. So if the regions is disordered, then we only using the sequence-related informations, do not using the structure ones. Okay, to this stage, we can conclude that our machine learning models can really learn the good structure alignment data with a powerful model as well as the well-defined features. So what's the problems remain here? Here, I just listed two. The first is the CFPRED only return the probability of the alignment instead of the log odds ratio compared to the background. That will make the result less significant, since we could not tell how goodness of alignment is compared to the random guess. The second problems of the previous method is we only incorporate the local informations on the alignment, while we don't incorporate the global information. What is global information? Because for the threading part, the template, we really know there are 3D structures. And for the sequence, we really can use some contact predictions, the distance predictions among the two residues. So if we can design a good algorithms to incorporate these global informations in our alignment model, we could boost our accuracies as well as the ability of the threading. So actually, our solution for these two parts is, first, we propose alignment potential with an elaborately designed reference states, and the model could be generalized into sequence structure as well as the structure alignment. Second, we incorporate both local and global terms in this alignment potential. For local term, CFPRED potential instead of probability is applied. For global term, the EPAD potential is employed to measure the two aligned positions' global fitness. So in the later slides, I will introduce these two parts in more details. First, intuitively, alignment is good as long as its probability is much better than the expected probabilities. That is also the basic concept of the log odds score. So this is actually inspired by the Blossom 6-2 matrix, as you can see here. The Blossom 6-2 matrix is actually a mutation potential for given two amino acids, A and B. The numerators can be regarded as the observed mutation probabilities given A and B, while the denominators could be regarded as the reference or expected mutation probabilities. So similarly, given one alignment A between sequence S and T, we define the potential of A as a similar manner that on numerators, this is the observed alignment probabilities, and on the denominators is the random guess or the expected. So in more details, we using these equations to represent the expected potentials. By that, to randomly sampled a few hundred, a few thousands of the random pairs x and y based on the constraints that x and y have the same length as s and t. So finally, we assume that the alignment is optimal only if it maximizes the potential instead of maximize its probability. Moreover, in order to incorporate the local and global terms together, the alignment probability given S and T could be modeled in a similar manner as previous CF-pred in a Boltzmann machine-like model with exponential families and potential functions here. So this equation is, you can get familiar with the previous CF-pred model. Now we use some simple math to derive our alignment potentials. Just to take the equations here and using the log, and we can derive this. The EXP here is the expectation operator that sum over all possible x and y. In order to make our algorithms possible, we just sample a few thousand alignments randomly in advance for the expected scores. The reason that we choose the x and y with the same length as s and t is based on the fact that since s and t has the same alignment space as x and y, so the partition functions of zxt and zxy differs only in mean values by definition, which is independent of any specific alignment, but may depend on the protein residue assumptions. Let's first model the local potentials here. From previous results, the local potential is defined as the f minus the expected terms. And we use a context-specific linear chain model as follows, which just using the same as talked previously. In summary, this will be only 9 constant values of e's here, because we have to do the summation of the random sampled alignments. All the previous trained parameters can be used without any changes in these equations. So here is slides for the differences between the maximize on probability as well as the maximize on the potentials. Because when we maximize on probabilities, we don't know the background. Yes, so we don't know each positions the background is good or not. So the generated alignment may be quite long, but with high false positive aligned positions. This is the drawbacks of the maximize on probabilities. While if we maximize on the potentials, the resulted alignment path will relatively be shorter but more significant, as well as more relevant to the background. So the maximize on potentials is very good for ranking the templates among the input databases. While suppose we have got a best templates, we can still using the maximize on probabilities in order to make the relatively longer path, which is required for the good models, which is built upon this alignment. So now let's go into the global potentials. So for EPAD, is actually a context-specific distance-dependent model to calculate the probabilities among the two aligned positions of S and Y. we can use these pictures for good illustrations. Suppose this is a protein structure template which is already known the 3D structures, and suppose the IJ is the two aligned parts here, and we know they are DIJs. And for given the sequence using the SI and SJ, we can use EPAT to predict their predicted potentials here. And suppose this predicted distance is highly fit with these DIJs, then according to these functions, we can say that the G functions is relatively high. So in one sentence is that the intuition underlying this equation is if the alignment is good, the distance of a sequence residue pair should match well in that of the aligned template residue pairs. So here we move to the result on the 1000 times 6000 data set. Here 6000 is actually the PDB 25 data set, which each of the pairs shares no more than 25 sequence identities. And the 1000 is randomly picked sequences from this set, and we do the threadings for each one against the 6000. And we exclude itself, of course. And then this is the result for the top hits. And compared with our new method compared to the HH-pred. From the result, we can say that the top model generated by the new CF-pred is much better, higher than the HH-pred. And also, if we compared our new method with the local potential used the CF-pred, we can also see that in the low structure similarity regions, which is measured by the TM scores, our new method compared the global informations is much higher than the local potential used. So finally, I welcomes you to using our servers. I will using some examples to show this. First, the server is our threading servers. Suppose we here is the new job window. You type the job name as well as you put the email address here, and you put your sequence here, and then you submit. You can get the result like this. So this example is actually a very long sequence with about 1,600 residues, and after our server run, you can see that we can decompose this original input sequence into 1, 2, 3, 4, 5 different domains, and the domain numbers you can see it on the return page by 1, 2, 3, 4, 5. Suppose we first click the first domains, you can see our predicted result here. We can don't let it spin, and this is the p-values which measure the significance. This is the predicted UGDT as well as the GGDT. You can see that the result is actually good, and also this is the alignment score. This is the template used to build the models. Also, you can show sometimes there are other more models in this, and the alignment is showing in these windows. For each aligned positions, we have made the local qualities for each aligned positions with the predicted RMSDs, while the 0 means the best and the 9 means the worst. So actually this domain is multi-domain templates, and for each domain we will predict the result. Also, the second purpose of our server is the bindings. Again, the input sequences will first be modeled as a 3D structures, and then this structure will be searched against a ligand database, and we will return the ligands put in the superpositions here. And you can see the pocket multiplicity indicates that how many templates with the ligands in the database share the similar structures with the input models. So the number higher here indicates the more reliability of the ligand seated in these positions. And also, we can show that there might be other possible ligands here. Finally, we provide another server which is just introduced by my supervisor Jingbo Xu on the contact map predictions. Again, the job is quite easy. You just type your email here, job name, and the sequence here, submit, and this will result in a contact map using our method Scicom. Yeah. Okay. So finally, I will thank my supervisor Jingbo Xu, my collaborator Jianzhu Ma and Feng Zhao, and this work is mostly done by Jianzhu Ma. And I thank ISMB 2003 and my institute as well as these fundings. And thank you for all of you participate. Thank you.
Okay, I think we're pretty much out of time, but in the changeover between talks, maybe we can take a question if anyone has a question. Can you tell me what method you use for disorder prediction?
Oh, disorder prediction, actually we use a third party, the Disoprad. Yeah, Disoprad 2. Yeah, we use there. Yeah.
Any final question? Okay.
Okay, thank you very much.